Okay, so I'm going to Zoom the fourth lab before your first exam. So first I'm going to go through the document, the lab exercises, and then I will go through the images. And then I'll be able to give you some examples of some questions at that point. All right, so let me just change my little share. Here we go to share. All right. So here we have Lab 4, Tissues and Integumentary System, Introduction to the Microscope. So we do want you to familiarize yourself with the parts of the microscope. You don't necessarily have to know what their functions are. The ones most likely to be asked of you are things that we use on a regular basis. And that would be the stage, your lamp source, your objective lenses, your coarse adjustment knob, the stage clips, the mechanical stage, fine adjustment knob. But to know all those is very, helpful, because if you need to move something, then you understand what you're going to be touching and moving. We're not going to, I will say that with this COVID environment right now, is that more than likely, I'm not going to have you use the microscopes. I'm going to put the histology on the board, on the screen at the front of the classroom. And then there will be some pictures, in the images within that lab for that week, that you will be responsible for. Okay, so when we get into the lab, you'll see what those particular parts are. You could always, if you have the lab book, that we don't require you to have, but you can, if you have that, you can look at pictures of it, as well as even in any textbook. All right, so here we have number 2, skin anatomy and accessory structures. Identify the tissues, quote unquote, structures and functions found in each layer. So the first part are the tissues. So when it comes to the skin, we have 3 layers, right? We have an epidermis, dermis, and hypodermis, right? So first what we want to do, and this sort of relates to how I approached this in the lecture class, where I showed you that picture of the skin. And I said, look, there's all this writing to the right and the left and the bottom. If you cover up certain parts of it, and just look at the kind of stripes of color, on this diagram, you had like a light tan on top, and then a pink background area, and then the yellow area below that. I said, start with these visible layers of something that you can see, and that would be the epidermis, the dermis, and hypodermis. So we're kind of using the same approach, especially as we apply it to the cookie sheet models, and then some of these are smaller little wooden models of the skin that are scattered throughout the lab. So we start with epidermis, dermis, and hypodermis. So what are the main tissues there? Epidermis is stratified squamous epithelial tissue, or epithelium. In the dermis, we have 2 major tissues. The primary is dense, irregular connective tissue, but we also find simple cuboidal epithelial tissue, because that is what the type of tissue that would form the ducts and glands of sweat glands, as well as oil glands, but you'll especially see the sweat glands, often clustered by themselves, not adjacent to a hair follicle. And then the hypodermis, we have areolar connective tissue and adipose connective tissue. And that's what will be written in there. Then it says identify the structures found in each layer. All right. So here we have sweat glands. I've given you the functions in there. All right. I could point to a sweat gland on a model, which I'll give you an example of in just a minute. And I can say identify one function, of that structure. You could say protect from infection or cools bodies. I could point to a sebaceous gland, and I say identify that gland, and you say sebaceous gland or oil gland. These are all like multiple choice or true and false. So I can say identify the function of that structure, and you'd say maybe protect from infection or lubricates the epidermis, keeps skin moist. Excuse me. And so forth and so on. A hair follicle, heat retention, protection from UV, touch reception, so forth and so on. All right. Free nerve endings, pain and temperature, reception or receptor. Meissner's corpuscle, pressure receptor or reception, light pressure, deep pressure reception, and I should be able to modify that. Right. heat pressure. And then we move on to the histology. All right, so then we're gonna look at some slides. Hopefully there's some pictures up there, in the images. I don't rely on those images so much. They're really a supplement to your studying and what you do in lab. But if there aren't reasonable examples of them in there, I will upload them this week. All right, so it says be able to identify the tissues from the slide. So you're going to be looking at a slide, and the pointer, might be pointing to a particular thing. It's like identify that tissue, or maybe identify the layer of the skin where you would find that tissue, something like that. So identify the tissues from the slides. For each tissue, identify its skin layer, and perhaps a function, which I just gave you up here. Okay, so we the layers that you're going to use are the answers, to the fill in the blanks here for the layers are all found here in bold print: epidermis, dermis, or hypodermis. So simple cuboidal epithelial tissue. In what layer would that be found? Technically, the glands and things are found in the dermis, right? And not that duct doesn't pass through the epidermis ultimately to the skin, but it's primarily found in the dermis. So that's what you're going to put in there. Function: forms glands. Stratified squamous epithelial tissue. What layer? Well, that is the epidermis. Function: protect from infection, dehydration, and UV radiation. So I filled them all in for you. You just have to memorize them. Dense irregular connective tissue. Layer: dermis. Provides strength and elasticity. Areolar connective tissue, found in the hypodermis or subcutaneous, helps anchor muscle to the skin. Adipose connective tissue, fat, found in the hypodermis or subcutaneous, stores energy, cushion, and insulate organs. Remember, whenever it comes to fat, you can always if you solidly remember the function of pure lipids from the chemistry lab, we keep saying long-term energy storage, cushion and insulate organs, then you're good to go. And that's really it for the document, but now we're going to look at the images. So I'm going to stop my share and reshare the images, and I have to toggle a little bit, hold on, back and forth sometimes. Let me go into this region. I just have to get into lab 4. And I'm trying, here's lab 4 tissues quiz, lab 4, there. Okay, now I can have something recently looked at so I can share it. There we go. There it is. Whee. All right, so let's start with 4-1. And let's see what we're looking at. All right, so this is an example of that cookie sheet model I always talk about. Here we have this pink with a little bit of tan on top. That's keratin. So you really have like the pink layer going across, then you have the peachy layer below it, and then further below that is a yellow layer. So that, which includes the pink that's undulating there, and above that layer, quote unquote, is the epidermis. And what tissue would I find there? Stratified squamous epithelial tissue. Right? Epidermis, stratified squamous epithelial tissue. Then we move a little farther down. Right? And below that is this pinkish, peachy, actually on this model, it's kind of a light peachy background. That region that undulates upward toward the epidermis or into the epidermis, that would be the dermis, right? What would be the tissues found there? Well, we just said you would have dense irregular connective tissue, right? And so that's not complete. You also need to include simple cuboidal epithelial tissue. Remember, these images there are kind of standard for a lot of, this should be meet the minimum requirements for really all instructors, but you always go with You know, the lab exercises and those sheets we fill out per class. Okay, then we have, lastly, that kind of yellow striped area, right? That's the deepest layer. That would be your hypodermis or subcutaneous. And we would find both adipose connective tissue and areolar connective tissue. So, you know, how might I ask a question? I could be pointing, having like a little arrow, you know, that has like an A on it. I said, you know, what is identify layer A? And you would say hypodermis or subcutaneous. Or I'd say identify one or the two tissues found in layer A, adipose connective tissue and areolar connective tissue. Okay, then we start looking at some of the structures that can be found on this model. Yeah, and even from this direction here, I'm just going to point to a few things. This over here, 9 in brown, and here in brown, right? Those are hair follicles or hair. Number 28, you see that with a little tail here, it kind of looks like a sperm. That's a Meissner's corpuscle. Over here, adjacent to the hair, here you can see it like 10, 11. Here's 11 over here. Those are the oil glands or sebaceous glands that use the hair follicle, the shaft of the hair follicle, to express oil to the surface of the skin. And then over here, number 21, these yellow tubes, you can see one to the right and left of this hair follicle. And if you follow it all the way down to the bottom, it kind of looks like a coiled hose. Those are sweat glands or sudoriferous glands. So that is pointing to the gland. That is a sebaceous gland or an oil gland. You know, what does it function to do? It, you know, secretes, its substance is an antimicrobial, helps, you know, protect from bacteria, it helps moisten the skin. You know, the functions I provided for you, something along those lines, it's fine. Then we have in the next image, dun, dun, oh, we're pointing to the little spermy dude there. That's a Meissner's corpuscle. What is its function? Light touch or light pressure reception. While we're here, let's just look a little bit over to the right-hand side. You see number 29, this little tree-like branching structure. It originates down here in the dermis, but extends into epidermis here, like branches of a tree. That's a free nerve ending. All right, so Meissner's corpuscle detects light touch or light pressure. Light touch and light pressure are the same thing. Okay, then we have, let's go to, there we go. Now we're pointing to, oh, that tree-like structure. All right, so structure and function. Free nerve ending, pain and temperature, or detects pain and temperature reception, or a pain and temperature receptor. All right, then we have, here's our sweat gland. So I said at the bottom, it'll be coiled kind of like a hose when you don't have it rolled on a spool of some kind. Right? So those are the sweat glands. Sudoriferous or sweat gland, regulate body temperature, acts as an antimicrobial as well. All right, then deeper down in the skin, here we like to use this example. Not that there aren't other examples here that you could use for a lamellated receptor, but this is the one we most commonly point to, right, for Epicenium corpuscle. Epicenium corpuscle. What does that do? Deep touch or deep pressure receptor. Okay, so that's our Epicenium corpuscle. Here we have another, we have some of these smaller wooden models. The cookie sheet model is actually rather large, like the mitosis model. So these are a little bit smaller, but we use them here rather frequently, as well as A&P too, because it's small and it's easy to transport from across the hall. from the A&P1 lab to A&P2. But of the things that I would say you should be aware of are the hair follicles. And I'm hoping, I'm not sure why he didn't point to hair before, and give you some pictures. They might be coming up shortly. So that would be a hair follicle and function, right? Touch reception, protect from UV, insulation. The oil glands that surround it. And then in white, instead of yellow, are some sweat glands here. Here we have some free nerve endings with a little bulb there. Those are the ones that I would most likely use as an example. Down here in the bottom, you can see the lamellated aspect to it. So it kind of looks like an onion that you cut into, and you can see all the layers. You know, that's not bad for a Epicenium corpuscle, but I'm more likely to just use those that are the most obvious. I think it is fair to ask you about layer, so that this upper layer, starting in blue and going upwards, you know, is the epidermis. The middle, with mostly tan in the background, would be the dermis, and wherever you see the fat and below would be the hypodermis or subcutaneous. All right, here's a slide of simple cuboidal epithelial tissue, right? Simple cuboidal epithelial tissue. Looks a lot like the picture from lecture. So identify that tissue, simple cuboidal. It's a little on the dark side. You know, getting pictures of these, and this was taken through the eyepiece, looking down at the specimen. When I do it and I upload them, I usually just take a picture of the screen in the front of the room. I think that's actually kind of easier, and it comes out generally a better light dark, you know, ratio, so you can actually identify a little bit better. But you can see these are cuboidal cells, and they look almost identical to the specimen, you know, of simple cuboidal epithelial tissue from this chapter in lecture. You know, what layer would it be found in? You know, in the dermis, primarily in the dermis. Then we have, yeah, this is why taking it to the microscope is a little challenging. This is a slide of all the skin. All right. Well, you don't quite get to the, you just barely get into the hypodermis. All right, so from this pink undulating portion upward, even though this is the keratinized region of it, from the pink undulating wall upward, you know, that is the epidermis. That tissue is stratified squamous epithelial tissue. That darkest staining point on the bottom, you know, that meets with the dermis then, or is adjacent to, right? That is that stratum basale, the area of high mitotic division for all those squamous cells above. Below that, where you see all this tissue kind of going in different directions, kind of looks like steak, that would be the dermis, right? And then farther down, you just barely start to see some adipose connective tissue there, so that would be the hypodermis or subcutaneous. Okay. Stratified squamous epithelium, the layer would be the epidermis. Okay, and then I'm going to scroll down again. Whee! All right, and so now we're pointing, right? That's the tip of the pointer. So whatever's at the tip of the pointer, that's what you're referring to, right? What layer would that be? The dermis. What tissues can be found there? Dense irregular connective tissue and simple cuboidal epithelial tissue. So remember, add simple cuboidal epithelial tissue to that. Then we have a pretty decent picture of adipose connective tissue. Again, it's hard to really get a good picture through the eyepiece of a microscope, but you can see this kind of honeycomb chicken wire appearance to this tissue. All right, now you know you're looking at fat. All right, adipose connective tissue. Hypodermis, so subcutaneous, and maybe a function, long-term energy storage, cushion and insulate organs. And this, it's, again, it could be focused just a little bit better, but it's hard when you're using your phone to take a picture like this. This is areolar connective tissue. All right, where do we find that? Subcutaneous or hypodermis. Function, you know, helps attach the skin to the muscle. That's fine. This is the one that I said is the kind of the Jackson Pollock painting of tissues. Has this random organization to it. Okay. And that's it for the images. Yeah, I don't know why. I think he forgot to put a picture of the hair follicle there. Or the hair, I don't know. But in any event, just know that you need to know the hair on that. I pointed, to those different structures, on the cookie sheet models in one of the first one or two images. So that's how you would apply the written material that we filled in for structure, function, layers, and apply that as much as possible to any model that you see. And it's, maybe what you should do is think about it as, okay, what am I looking at? What do I need? What do I need to know about this image, or this symbol, like a Meissner's corpuscle? Well, it's a Meissner's corpuscle. It's found in the dermis. It functions as a light touch or light pressure receptor. Let's see what else. That's really about it. Okay, so I'm going to stop this share, and we will resume in class and, look at as much of the tissues as possible in there. And okay, that's it.